Hey, how's it going, everyone? I will be talking about embryology. This correlates to chapter 6 of the Brain Facts book. However, I have added some additional info from other sources, and if you guys would like to know more about it or just cross-reference things, because a lot of these things are relatively new developments, so if you guys want to read more about it, then I'll have a reference sheet at the very end. But for now, let's talk about the embryology. So this is just a small chart talking about really the chronological order of the entire development of the future child, right? In the course as the brain bee, we are going to be talking about the central nervous system, right? This big portion right here. So let me change this slide real quick. So we'll be talking about this section right here. This is the main focus for our discussion. And as we go through the presentation, we'll actually even be going into some part of after birth, right, for myelination and whatnot. But something that I really just wanted to show you guys, or really bring to your attention, is the fact that the CNS starts developing extremely early, before 3 weeks. It's just that the chart isn't long enough to show it. But the issue with this is that sometimes there are harmful things that we can do to the embryo and just because the CNS develops so early, a parent just does not know they're pregnant. So, and we'll talk about that later on when we get to it. Moving on. Immediately after fertilization and blastulation, we have a process called gastrulation. So gastrulation, by definition, is just the formation of the trilaminar layer, which is the ectoderm, mesoderm, and endoderm, which is derived from the epiblast. So we have to think, well, how do we go from one layer, the epiblast, to 3 layers, ectoderm, mesoderm, and endoderm? Well, the first step in this process is the formation of a primitive node. So basically, a primitive node is just a little divot kind of that forms in the center of the epiblast plate, and then a primitive streak forms, which just extends that little divot caudally into a long line from the node. And this whole process is initiated by the molecular signaling factors called WNT and activin. Now, the reason why these things form is because what will happen is cells will actually migrate from the epiblast plate into this primitive node and primitive streak and form inward, and then it'll eventually form 3 layers. So what it has something called cilia. So if for any of you guys are taking physiology, we have cilia in our esophagus and our GI tract to help us push food along, or the bolus of food. Instead of a bolus in this case, the cilia on the primitive node will actually push the cells into this primitive node and primitive streak. And then once the cells migrate to their location, they will form the mesoendoderm layer. And then from here, nodal, which is another signaling protein, will activate chaperone proteins such as GATA, SOX, and FOX, which will then help differentiate this mesoendoderm into the mesoderm and then the endoderm. And then finally, at the very end of the process, the notochord forms. The process of the formation of the notochord is that the notochordal process will develop on its own, then it'll integrate with the endoderm, and then separate to become its own notochord. Now, the notochord is extremely important in the formation or the neurulation, as well as the morphological shape of the cells. We'll talk about later. This is something that actually deviates a little bit from the Brain Facts book, because all of the very current data that I've collected shows that it's the notochord that does it, whereas in the Brain Facts book, it says that the mesoderm does this. But just because of all the data I've collected on this and the research papers I read about this, it says it's a notochord. So this is a small deviation from the book. I just wanted to let you guys know if you read it and get confused. So this is a little picture showing the epiblast plate, right? This whole thing is epiblast plate. And this little red circle is a primitive node. And this little streak down here is the primitive streak. And these arrows show that these cells are moving inwards. This is really showing what the previous slide, but just in a more video format. So I'll actually play while I'm talking. But basically, the red dye shows the anterior primitive streak cells, which will form the periaxial mesoderm. This is a precursor to somites, which is the precursor for the axial skeleton. as well as the dermis, the muscle. And you can even see from this video that even so early in the embryonic development, you can see the almost a little bit of a the vertebral processes forming already. Very interesting. Now, this green dye that's forming is showing the posterior primitive streak cells, and this forms the lateral mesoderm. This includes the skeleton of the limbs, as well as the muscular wall of the GI tract. And you can kind of see there's a little gap in between the red and green dye. This is the intermediate mesoderm, which will give rise to your gonads, your kidneys, and as well as the urogenital tract. So I think the real question is how is it that a group of cells, like the epiblast layer, how is it that this very organized group of cells is able to spontaneously create new cells that aren't organized and allows it to follow the cilia, right? Well, this is a big concern because this is analogous almost to anchorage dependence proliferation. For those of you who have studied a little bit of cancer biology, this is a very big indicator of cancer formation, wherein cells will normally they have to be anchored down to a membrane of some sort. Usually the extracellular membrane. It has to have attachments to it to proliferate. But in the embryonic formation and the proliferation of these cells, we see that we have an epiblast plate which is very organized, but then we also have cells that are proliferating that are disorganized that are able to move wherever they need to be moved to form 3 layers. But the question is, how does it do this without being considered cancerous, right? How does it do this? How does it do this in a healthy way? So because of this problem, or because of this issue that scientists have encountered in their theories, was the epithelial mesenchymal transition, or the EMT. So what the EMT is a very highly controlled process where the epithelial cells, or the very organized cellular layers, become or transition to the mesenchymal cells, or the very disorganized cellular layers, and vice versa, which will then allow for cell migration and proliferation. And then, so for example, in the gastrulation, the epiblast layer is epithelial cells. It goes through this EMT to become disorganized cells, and then once it forms the trilamellar layer, the ectoderm, mesoderm, endoderm, then it goes back to epithelial cells in these very organized cellular layers. And the way that this happens is, I added this picture, kind of show this, but one of the things that you lose, right? So this is actually in a chicken gastrulation, right? This is how we've hypothesized that humans have this as well. This is a model after chicken gastrulation. But basically, so the transition is medial to lateral. So you guys can see in this picture, these medial cells are actually becoming mesenchymal, whereas these lateral cells are still epithelial, and in this middle region, they are transitioning, right? So one of the biggest things that we see is the loss of a basement membrane. Additionally, these microtubules are pushed back a lot more, so they're closer, and so there's less centromeres in the cell. You can see the stark difference between these mesenchymal cells and these epithelial cells. Additionally, you can also see that there is no basal cortex at these mesenchymal cells, whereas in these epithelial cells, there are. Cool. So we talked a lot about the formation of notochord. Now, one discrepancy that I found within the book and current literature is that it says that neurulation, which is something that we'll talk about in the next slide, is induced by the mesoderm. However, in the current literature and everything that I found, everything contradicts that and says that the notochord actually induces neurulation. So if you guys are reading the Brain Facts book and you see this presentation and you guys notice this discrepancy, that's why. And of course, like I said, I'll have the reference sheet at the very end. So if you want to look at it yourself, you can do so. But for the notochord, there's 2 main functions. 1, mechanical, and 2, morphological. So the mechanical function is to influence the folding of the early embryo. This is called neurulation, and as I said, we'll talk about this later. The other is the morphological function of the notochord, which is through the secretion of Sonic Hedgehog. So yeah, so this is named after the video game character because so typically in the scientific world, Whoever discovers a new molecule is able to name it whatever they like. However, the scientist who discovered Sonic Hedgehog didn't think it was very important, so he thought he would just name it after a video game character because no one else would ever really learn about it. Little do you know, it is probably one of the most important molecular signals in the CNS and in embryology, so at least it's easier to memorize now. However, Sonic Hedgehog is very interesting in the way it works because depending on depending on the concentration of Sonic Hedgehog in the environment, a stem cell will become different things. So in this example I have here, the cells with the highest amounts of Sonic Hedgehog will become specialized glial cells. And then cells with less Sonic Hedgehog will become motor neurons. And then cells with very little Sonic Hedgehog will become interneurons. However, if you pay attention to the vocabulary that I'm using here, I'm not using the concentration of Sonic Hedgehog, I'm using distance from the notochord. The reason for this is a little bit hard to explain, but the best way is through an analogy. So let's say that you drop a drop of dye into a cup of water, right? Now, the point of contact between the dye and the water is, we'll call it the origin of the dye, right? And the further you get from the origin of that drop of dye into the water, the less dye you can see in the water, the less discoloration there is, right? The reason for this is because the farther out you go, the more volume that the dye has to take up, and there's just a less concentration of it, right? So take from that analogy, we can make the association that there's a correlation between the concentration and the distance from the origin. Now, in this example, Sonic Hedgehog is going to be the dye, and the notochord will be the origin. So in this case, it is the distance from the origin, which will, or the distance from the notochord, which will determine what the cells become. So cells closest to the notochord will become specialized glial cells. A little bit further away, they'll become motor neurons, and then even further away, they'll become interneurons. Now the notochord has another function, right? So it is also forms the nucleus pulposus, which is inside of your intervertebral discs. So the intervertebral discs is a disc in between each vertebral body, right? And a little clinical tip for you guys, a little clinical correlate for you guys is that if you've ever heard of someone herniating a disc in their spine or something, it's because this nucleus pulposus had bulged out of where it's supposed to be, inside of the vertebrae, right? The reason why it will oftentimes compress a nerve is because there's two components to the intervertebral disc. One is this jelly-like substance called the nucleus pulposus, and then a second harder casing called the annular fibrosis. Now, these herniations, these disc herniations, occur posterolaterally, right? So if you think of like a clock, posterolaterally can either be 4 to 5 o'clock or 7 to 8 o'clock, right? Behind you and to the side. Now, because the annular fibrosis is very thin posterolaterally on either side. And posterolaterally to the disc is your intervertebral foramen, which contains your spinal root nerves. So if you have a disc herniation, that there's a relatively high chance that you can compress a nerve and cause sensory damage or even motor damage to that nerve and the muscle innervates and whatnot. So we've already talked a little bit about neurulation, but as we already talked about in the previous slide, right? Neurulation is induced by the mechanical function of the notochord. So the ectoderm, which is the outermost layer of the trilamellar layer, when it is signaled by the notochord, it will begin to thicken in the middle and it'll form a neural plate, right? And then it'll also form neural crests adjacent to this plate. Now, what will happen is this neural plate will begin to fold and it'll form a neural groove, right? Little groove. And then it'll fold further and form a neural tube. And then at the same time, the neural crest cells will then separate from the plate and become its own structure. So a little piece of paper here to show you guys. But piece of paper, this will be the neural plate. What happens is it'll form a groove, and it'll eventually close over into a neural tube right here, right? And then like I said, the neural crest cells will separate from the neural plate before the tube is formed. And now this neural tube will then go on to form the rest of the CNS. And the neural crest cells will then form the DRG, the dorsal root ganglion, which is important for sensory and afferent information, as well as connective tissues in the head and neck. So just to give you guys a nice little visual about what we're talking about here, right? So this blue, so this is kind of in the middle of neurulation, but this blue is ectoderm, and you guys can see how the neural plate is formed, right? Is thickened and forming the neural plate, and then adjacent to it, we have these neural crest cells. And the notochord is down here inducing this whole process, as well as the mesoderm, which is the layer underneath the ectoderm. And then it'll induce it to start to fold inward, and you see the neural groove is formed here. These neural crest cells are still attached. Now, further folding and fusing of the edges will form the neural tube, as well as the separation of the neural crest cells. So at the end of this process, what we have is we have the whole, the ectoderm, or now it's called the epidermis, right? Then we have the neural tube, we have our neural crest cells, we have our mesoderm, which are somites. And if you guys remember, the periaxial mesoderm became the somites, which then became the vertebrae, as well as we have our notochord, right? Cool. So now that we have talked about how it folds, one thing to mention is that the neural tube will fold and fuse in the center first, and then over time, over the next few days, it'll begin to kind of zip up as well as zip down. And eventually the top end, the head end, or the cephalic end will fuse, and the caudal end, or the bottom end, will fuse. Now, the cephalic end of the neural tube fuses at day 25, whereas the caudal end will fuse at day 28. And then through numerous cycles of proliferation and migration, as we'll talk about in the future, after a little bit of clinical slides, you'll eventually form these bulges, which is the forebrain, which is your cortex, some of your limbic system, your midbrain, which becomes like most of your basal ganglia, corporal quadrigemina, red nuclei, etc., as well as your hindbrain, which is your cerebellum, medulla, and your pons. And eventually, after proliferation and migration, you will eventually form the child's brain. But now we've talked about the folding, the process of neurulation in the neural tube, let's talk about some pathologies associated with this, right? Some clinical correlates. So one of the things that can happen. So this whole slide is dedicated to if the caudal end of the neural tube fails to close, right? At the bottom end of it. So all these pathologies are associated with the lumbar spine, right? So what will happen in this process, in the spina bifida occulta, is that the nervous system is completely intact. There's no pathologies, nothing wrong with it. And in fact, it's actually covered by the postvertebral muscles. The only issue here is that the vertebral arches didn't form properly. So what will happen in this case is there's really no visual symptoms. It's actually symptomless in terms of the nervous system, but one of the biggest hallmarks of spina bifida occulta is that the child will have like a patch of hair around where the spinous process didn't form. And this is symptomless, so the child will probably live a completely normal life, no surgery needed, nothing required. Now, the next is something called a meningocele. So if you don't really want to brute memorize all these definitions, right, this one and the following ones, one thing you can do is you can kind of just look at the roots of the word. So meninge in meningocele means the meninges, right, the covering of the brain, the dura, pia, and arachnoid. Whereas cele means just a swelling or an outpouching almost. So based on its definition, from these roots, it is the outpouching of your meninges. And now naturally, within your meninges, right, between the layers is CSF. So this will present as a cyst filled with CSF, usually in the lumbar region. And the nervous system is usually intact, so this is removed surgically and is not too bad. Very good prognosis for the child when it's born. The next definition, or the next term we want to talk about, is a meningomyelocele. So meninge, we talk about meninges. Myelo actually means spinal cord or nervous tissue, and cele is the outpouching of the swelling. So this is similar to a meningocele, but now the cauda equina, right, the nervous tissue, is now within this outpouching sac. Now, the biggest issue here is that the nervous tissue is historically shown to be adherent to the sac, so this will have to be treated surgically, but very carefully because you don't want to break or cut a nerve. Now, the next thing that we'll be talking about is a myelocele, right? So this is similar to a meningomyelocele, but instead of there being the meninges, right, as is shown by the name, it's just the spinal cord outpouched, a swelling of the spinal cord outside of this body. So basically, this condition is, so the spinal cord will lay exposed out of the infant's back. And as you could probably deduce, this is very bad. So typically the child will be stillborn, or they'll be born dead, or they will shortly die afterwards due to infection of the spinal cord. So at the bottom here, we can see that there is a syringomyelocele, right? I think the best way to really talk about this is really compare and contrast this with a meningomyelocele, right? So as you guys can see here, this is a pretty normal spinal cord, except obviously for the fact that it's not here. You're really close to spinal cord or the vertebral column. But as we can see here, we have these gray matter tracts, right? These gray horns. We also have these white columns, and in the very center, it's so small you can't see in the picture, but there's something called a central canal. It is continuous with the ventricles in the brain and actually allows CSF to flow through. However, in a syringomyelocele, that tiny little pinpoint of a hole becomes a very massive hole. And if this is too big, then that means that there's less room for the spinal nerves and ganglia to form, which can cause severe nervous system damage to the child. So paraplegia, loss of sensation, motor movement in the lower limbs, etc. You see here the myelocele. There's almost now the spinal cord is basically almost outside of the body. This is how infection can be formed, right? It can directly get in contact, bacteria can get in contact with the spinal cord, and a lot of issues can take place. And spina bifida occulta, pretty normal how the entire spine should be, except that there should be a bony process right here called the spinous process. Forms almost like a triangle or a curved triangle almost. The meningocele, as I said, is just the meninges, then a CSF cyst that's forming outside the body, and that's about it. Cool, so now we've talked about the pathology associated with the caudal end of the neural tube. Let's talk about the cephalic end, as well as the treatment for pathologies associated with neurulation. So there's really one main thing associated with pathologies of the cephalic neural tube folding, and that's going to be anencephaly. Now, of course, there's going to be many other pathologies, but this is the one we're going to focus on, right? So anencephaly is a very devastating pathology because the majority of the brain, the cerebral cortex, and the majority of the skull vault is going to be missing, right? And just caused by the failure of the cephalic end of the neural tube to fold properly. And as you guys can probably guess, right, most infants are stillborn. Now, how do we prevent this, right? What is a treatment for neural tube folding pathologies, right? Well, in many studies, it was shown that taking a folic acid supplement, about 4,000 milligrams, has been shown to dramatically decrease the rates of a neural tube defect, caudally or rostally. Now, clinically, right, in the real world, the issue is that this process of neurulation, right, if you guys remember, it finishes before the 28th day, before the first month. And a lot of the times, the female, the woman doesn't know that she's actually pregnant. And she doesn't know she's pregnant, that she doesn't know to take a folic acid supplement. So as a result of this conundrum, right, physicians strongly suggests that all women who are capable of being pregnant consume at least 400 milligrams per day. So now we talk about neural tube folding, right? The neural tube is a very tiny structure, right? It's very small, centimeters in size. So what we have to do now is we have to grow. We have to proliferate these neural cells. However, in the beginning of the process, right, when the proliferation is starting to occur, it starts from a neural tube, but it's so small and there's very few neural stem cells and progenitor cells. So the response to this that the body has developed is a biphasic approach to proliferation, right? The first stage of division is something called symmetrical division, wherein if one neural stem cell or progenitor cell divides, it'll create two identical daughter cells that can then keep on dividing themselves, right? Now, once it reaches a certain amount of dividing cells, then it'll transition to the second stage called asymmetrical division, wherein when these cells divide, it'll create one daughter cell that can keep on dividing, but it'll create another cell that can then progress towards its terminal cell type. So like a glial cell or something of the sorts, or a neural cell. And as I said, this is a biphasic approach. And as a result of this, right, you start with very few neural stem cells, but you also end with very few neural stem cells and progenitor cells, because the second stage makes sure that that there isn't an excess at the end of the stage. Now, the few that are left will go to areas of the brain involved in memory, such as the hippocampus, some areas of the frontal cortex, etc. Now, what if this process goes bad, right? What happens if too many daughter cells are created, if this first stage goes on for too long, or if the first stage goes on for too little of time, right? Then that can cause microcephaly or megalocephaly. So microcephaly is caused when the first stage of division doesn't occur long enough, and this can be an issue with the molecular signals that facilitate these processes. A megalencephaly can be caused by the first stage of division, the symmetrical division, occurring for too long, again by issues with the molecular signals. Now, why is it that, so as a result of this, the brain will either be too small or too large, right? Now, why is it that, when you think of the skull, it's a closed skull vault, right? It's a closed thing. So how is it that if a child has a smaller brain, then it will cause a smaller skull and a smaller head? Or if you have a bigger brain, it'll cause a larger skull and a larger head. Well, that's because unlike you and I, right, a child has their skull is in pieces, right? So they have an anterior fontanelle and a posterior fontanelle. And their skull is more flexible than ours because it hasn't ossified and fused at the sutural linea. So as a result, it's very flexible in terms of it can grow smaller or grow larger depending on what area or volume it has to cover. This can result in children with actual small heads and children with very large heads as a result. So now we've talked about proliferation of these neural cells. Now we have to talk about the migration. How do these cells go from one area to another, right? Now, proliferation occurs in the neural tube at the ventricular zone. You know, it's called the ventricular zone because it is the inner edge of the neural tube. And for those of you who didn't know this, but the hollow end, so it is a tube, and in the center there's a hollow space. This hollow space will eventually become the ventricles. So the inner edge of this neural tube will become the ventricular zone because it is the zone closest to the ventricles. So the goal of migration is to go from this ventricular zone to the marginal zone, which is on the very outer edge of the neural tube. Now in studies, it's been shown that 90% of these neurons are guided to their final destinations via radioglial cells. Now, radioglial cells are formed in the intermediate zone, right? This is the zone between the ventricular and the marginal zone. So the innermost zone is a ventricular zone, the middle zone is the intermediate zone, and the outermost zone is the marginal zone, right? Now these radioglia act as scaffolding for these new neurons to move outwardly radially, right, towards the outer layer. However, there are certain teratogens, such as alcohol, cocaine, or radiation, which can prevent proper migration of these neurons. Now, it just really goes to show that these neurons, if they don't go to the correct location, the brain can't function properly and can even dysfunction, which is why intellectual disabilities or epilepsy can form as a result of uh, altered or messed up migration of these cells. So as we talked about throughout this video is neurulation, proliferation, and migration, right? The difference between this and what we're going to talk about now is that these three processes, neurulation, proliferation, migration, are all internal processes, right? They happen regardless of what's happening to the child, right? Unless you have some teratogen which can alter these processes. But for the most part, it's internal and it's independent, right? Unless some very strong external stimuli affects it. However, now that these neurons are in place, these cells are in place, now you make connections, right? And this is influenced by the external environment. So even very early, even when the embryo is still developing, the fetus is still developing, these connections are made when they hear their mother's voice, when they feel the heat being inside the womb, when certain smells, right? They hear different things. These all forms connections between these neurons, and it's all due to the external environments. And we'll talk about later, if there is no external stimuli to for these neurons to fire off, then they'll actually be destroyed. We'll talk about that later, very soon, actually. So how do these connections between neurons occur, right? Well, they occur via dendrites and long axons. Right, dendrites will receive signals, and these long axons will transmit signals. Now, these long axons are very interesting because they can actually grow to be over a meter long. However, the neuronal cell body, the soma, is very small. So it's been shown that the axon can be a million times longer than the cell body, because there's some axons that go from the spinal cord down to your toes, right, for motor or sensory innervation. So in tall people, this can be over a meter long. So how is it that these axons can get to how is it that the axons can get to this region, right? How is it the axon is able to perfectly synapse where it's supposed to be at? And their answer to that is that the axon is formed due to a growth cone, right? So a growth cone is something that the soma will kind of grow, and this growth cone has receptors on it. And the job of these receptors is to find cytokines or molecular signals that it's attracted to and go towards it, and avoid other molecular signals that it doesn't like. So in the context of this example, these growth cones will use their receptors and look for the cytokines that their target is secreting, and its main job is to find the cytokine signal and then go towards it so that the concentration increases, right? Now, if we go back to our example of the notochord and sonic hedgehog, right, the closer you are to the origin, the higher the concentration of the signal. So in this case, the higher the concentration of the cytokine, the closer you are to the target, right? So that's how it's able to very accurately get to where it's supposed to go. And some of these molecular signals include netrin, semaphorin, and epherin. So synapses, right? These connections, are they actually connected? The answer is no, actually. A synapse is a very small gap, about 10 nanometers, between the axon and the dendrite. And what happens here is in the synapse, it is a conversion of electrical signals to a chemical signal back to an electrical signal. So what's going on is that the axon will transmit information, right, based off of an EPSP, an excitatory signal from its helic, which will cause a depolarization of its membrane. This membrane depolarization will then go towards the presynaptic terminal, which will release something called neurotransmitters, the chemical, right? So this neurotransmitter will then go across the synaptic cleft and bind to the receptors on the postsynaptic membrane, or the dendrite, which will then cause, depending on if it's excitatory or inhibitory, depending on the neurotransmitter, which will cause a electrical difference in the dendrite, which will cause it to go and move via saltatory conduction towards the soma. When this neurotransmitter binds to the postsynaptic membrane of the dendrite, then it'll induce some kind of electrical depolarization, assuming it is excitatory, then which will then allow it to go to its proper destination. And the intermediary between the synapses, I just said, neurotransmitter, and the reason why our brain is so good at processing information so quickly all at once, and very complex information actually, is because we have trillions of these synapses, and there's thousands of interconnections between neurons between each. But for this to be efficient, right, for this to be an efficient process with so many axons and synapses and neurotransmitters to use, how do we make sure that it stays specific, right? How do we make sure that uh, that the axon releases a certain neurotransmitter and the dendrite is able to respond to that neurotransmitter? And there's three main ways. So the first way that these neural connections has specificity is one just based on where the axon goes, right? So if you remember, the initial growth cone is very specific to a target cytokine, and typically the target will respond to a specific cytokine that the growth cone or the precursor to the axon will have. Secondly, when the axon and the dendrite come in contact with each other, the axon will become specialized for a certain neurotransmitter, and the dendrite will react to the neurotransmitter released and develop certain receptors that can respond to it. And thirdly, in recent studies, it has been shown that astrocytes are able to even facilitate and regulate synapse formation. So in the paper that I found on PubMed that allowed me to write the third point was describing a study where there's two petri dishes, right? One with a whole bunch of neural cells, and the other one with neural cells, but also with astrocytes. In the one without astrocytes, only very few synapses were formed, whereas in the petri dish with the astrocytes included, there was many thousands of synapses formed. So it showed to the researchers that there is that it plays some role in synapse formation. So now a quick little section about myelination, right? So I'm sure you guys have learned this already, but the function of myelination, right? Axons are myelinated for the ability to have saltatory conduction, which will very quickly allow for electrical impulses to travel throughout the axon. The reason being is if there was no myelination, right, then you would have to depolarize every tiny segment of the axonal membrane. However, with saltatory conduction, you only have to depolarize the membrane at these nodes of Ranvier, and it drastically increases the speed of conduction. Now, myelination, of all these processes we talked about previously, it's always happens during development of the future child. But myelination actually occurs throughout your lifespan. If you guys know, like when a child is born, they have certain reflexes, right? Like they have a grasp reflex, they have a Babinski reflex, right? And these are evolutionarily developed. But these are only reflexes from the spine, right? These are interneuron reflexes. The reason why the reason why these reflexes are present but then eventually disappear is because the axons from the brain down to the nerves are myelinating, right? So before the myelination, when the baby's just born, before there's myelination, these axons, these connections, these signals dissipate before it has a chance to communicate. But once it myelinates, then these reflexes disappear and the baby loses their reflexes or the reflexes that it had at birth. Now, the CNS is myelinated by oligodendrocytes and the PNS is myelinated by Schwann cells. Now, pairing back, right? So I've already talked a lot about proliferation and migration. However, if you guys, this is a very complex process, right? With a lot of moving parts, a lot of neurons, and there's a lot of movement, right? So because of this, there just naturally leads to a lot of redundancies and inefficient neurons. So as a result, through the pairing process, the pairing back or the pruning process, about half of the neurons that are initially created are actually destroyed by apoptosis. And apoptosis is programmed cell death. Now, apoptosis will occur if the neuron doesn't, one, get enough trophic factors, or two, is just not being used, right? So an example of a trophic factor is that a specific one that's essential for the survival of sensory neurons is nerve growth factor, right? So nerve growth factor has to be present for these sensory neurons to survive. And I've already talked previously about if a axon or a neuron isn't being used, if there isn't any electrical signals going through for some time, then it will be apoptosed, right? This is a literal example of if you don't use it, then you're going to lose it, right? This is how, yeah. So now one of the very interesting things I found, especially in neurodegenerative research, is that apoptosis is actually held check in adulthood only by a reduced amount of trophic factors, right? So when you're developing, you have a whole bunch of trophic factors, but as you grow, you lose a lot of these trophic factors to a point to levels where it's just meant to maintain. But this is a hot topic now in neurodegenerative research today, because this may explain why some certain regions of the brain just die, because we don't know the actual mechanisms and why, but this may be an answer to some of those. So this is highly researched currently. Now, something else that I found is, of the many functions of the astrocytes, right? Another function of it is that it has been shown to aid in the pruning of these neural cells by phagocytosis. So pairing back, just to summarize, is done by two processes. One is by apoptosis, and the other is by astrocytic phagocytosis. Now, this is the reference to slide. So if you guys click through any of these links, then you guys will be redirected to research papers, different books that I used to add upon the Brain Facts book. So I hope you guys learned a good amount. If you guys have any questions, be sure to ask us, so see you guys later.